Prem, shall we go ahead?
Yeah, please.
Okay. So, thank you, Dr. Prem, for joining us. It is my privilege and pleasure to introduce Dr. Prem Singh Kaushal. Dr. Prem obtained his PhD from the Indian Institute of Science with Professor Vijayan. He then did two postdoctoral fellowships, one at the Case Western Reserve University and another at the Biggs Laboratory Wadsworth Center. Following this, he joined the Center for DNA Fingerprinting and Diagnostics in 2017, where he was leading a research team. He then subsequently moved to the Regional Center for Biotechnology, Faridabad, where he is currently positioned. His research focus has been in unraveling the translational or translation structure biology, if I may call it, primarily focusing on protein synthesis, regulation, and ribosome biogenesis in pathogenic microbes such as Mycobacterium tuberculosis and Entamoeba histolytica. And he extensively uses the Cryo-EM technique in his works. He is the proud recipient of the DST SCRB Early Career Research Award in 2019. I must also add that his YouTube video on opportunities and fellowships in India to pursue a career in research is a must watch for all young aspiring students. With this brief introduction, I welcome you again for this online seminar series. Dr. Kaushal, stage is now yours.
Okay, let me share the screen first. I think I'm audible and my screen is visible.
Yeah.
Okay. Thank you, Dr. Bigneshwar, for kind word of introduction, and I want to thank you for as a organizer also, and thanks to Dr. Tagesh for giving me this opportunity to talk in front of you. So basically, from the morning, previous speakers has set up a very nice stage or platform. And I think without much difficulty, I can just move on to my the topic that is assigned to me, that is the Cryo-EM data processing, right? So if I can take that fancy cursor, just one minute. Just a minute. Laser pointer, yeah. So data means already this portion is covered very nicely from sample preparation, then data collection, etc. So most likely, and we are here once you get a tons of or terabytes of data, and how to reach from here to here. I'm going to talk very simple on this step. Bits and pieces, this portion is covered, so that will be kind of a revision. And feel free to interrupt me, or you can note down if you have any question. Let's move on. See, from Cryo-EM, you get a terabyte of data, and you have to apply have to be smart enough that you handle this data nicely and get a high resolution structure. So what I'm going to talk is first tell you the basics of image processing. Then I will take as an example that we are currently using and how we have successfully used to determine the cryo-EM structures in our lab. Okay. The problem is with biological material that we know that, see, there is a radiation damage. So we collect data at low dose. And because of low dose, all cryo-EM images, they comes with a lot of noise. You will see sometimes hard to see the particles, and that's kind of data sometimes you have to deal. The another problem is because we cannot collect biological material data at the focus, we have to go away from the focus in the deep focus to add the contrast. And in electron microscope, these electromagnetic lens, they are not the ideal lens. There is the artifact. So that creates a strong artifact that we have to address by CTF, contrast transfer function correction. And another problem is because particles, they are fallen randomly, we don't know the orientation. Basically, this 3D image processing is to find out the right orientation of that 2D projection. And we don't know which class it belongs, right? Let's try to understand how this is focused. This single particle reconstruction is mostly developed by, I must say, by Joachim Prem, the Nobel laureate of 2017 in chemistry. See, to deal with the noise or noisy images, one way is you add up. See, if you have one noisy image like this, 4 will give you a lot of some feature. If you keep on adding 30, 250, 1200, 9000, you get very crisp, clear images. Ideally, this is the concept. That's why you need thousands of your particle or hundreds of the microgram. And just to summarize, to give you an overview, this EM single particle reconstruction is nicely elaborated here. It's from the Nobel lecture of the Joachim Prem, that from the announcement of the Nobel Prize. Basically, what in your microscope, these are the particles, they are tumbling in ice, and what you get is the 2D projection. First thing is you try to align 2D, stack them, that particle, they have fallen nearly in the same orientation, right? These particle has fallen like this has fallen like this. So you stack them, then you get very crispy, the signal to noise ratio goes up, and you could clearly see, okay, this is one projection, second is second like this. Then there are different methods computationally. Ideally, you use a low resolution map and back project them and generate your 3D construction. That is the simple image processing is, and it was successfully applied first time for large subunit of ribosome in 1987. This is a negative staining because by that time the cryo was not developed yet. Okay. So there are different steps that we go in image processing. First is we get movies, so we do motion correction, CTF find, manual picking, 2D auto picking, then 2D, 3D, movie processing, polishing, how to treat the heterogeneity, auto refinement, post processing and refinement. Let's go one by one what we do in basically while going through all these image processing stuff, right? Okay, so this is rely on regularized likelihood optimization software, one of the most widely used software. There is Cryo-Spark is also, but the concept is more or less same, okay? Whether you use rely on or the maximum likelihood or the Cryo-Spark. What happens when you are imaging your sample in an EM, when the electron falls on your eyes, it whirls. Exact mechanism is not known, but it whirls like this. So what is happening when you are imaging, your particle is basically drifting, right? And if you expose for too long, particle are drifting, you will get a blurred image. So the first step, we collect data in a movie mode that I'm sure all of you know by now. These are the movie frames and they are drifted. First thing we do is we apply a drift correction. It is a simple, like these days, the smartphone are there and you want a very nice crispy image, you apply this de-blurring or something if the object is moving, right? Simple thing that is called movie motion correction. And this was first time successfully used by et al. With Shreyas Shore's group. And you can trace out, once you apply the movie correction, you can trace out how that particle has drifted and apply a correction and put it back. This is based upon you have segmented your micrograph in small section and then applied and corrected. It is a rough correction, not the, I must say, not the fine correction. So this way, now second comes to the CTF correction. CTF, as I said, because there are a lot of artifacts, what you do, you apply a mathematical correction, right? You have an image, you do this fast Fourier transformation, you get these thorn rings. This is from the experimental image. You apply a correction and it should match. The more the thorn ring you see, more it has the information. Okay. And when you are selecting based upon your thorn ring or CTF, you can see there is a ice. Maybe this micrograph has a ice. It may not be good. And this also has a ice, this has a ice. So you need initially to manually check and see after doing motion correction, manually check good particle, check for good CTF. Ideally, it should look like this. If you see like this, then you see your micrograph, whether that ice is throughout or it is in some corner, whether it can be used or not. Okay. Now, next step is once you have corrected, then you do a manual picking and alignment. You pick particle like this, and like this, where you find. So the problem is here because experimental images, I said that they are random and their orientation we don't know. So we have to find the orientation. In term of Shor's term, we call it Shor's term. We call it as a missing data. To suppose if each image is come a tag of orientation also, then there is no problem. The data processing is automatically solved. But it is not there. So what we are trying to get is our 2D particle or our particle trying to find out the best alignment or best orientation in our model. So if it could have been with some tag, this is the orientation, you could have got the fine penguin. How to deal with this? You take a reference image that may be some low resolution or blurry image, and because our eye, we can see that, okay, this penguin is not matching with this, so it needs a rotation, but computer doesn't understand that. So you keep on rotating like this and finding the cross correlation function with the rotation at this angle of rotation on XY plane. It is finding highest cross correlation function. So you assign this angle or this orientation as a for itself. This is the image alignment procedure. And this is when you are using this least square method. And another method that is the maximum likelihood. It is same in crystallography also. We use least square and maximum likelihood. RatMac use maximum likelihood and CNS use least square method. I don't know at the present, but at that time it was the same. So when you are using maximum likelihood, you do the same procedure, but you assign a probability tag that it has the, say here it has the highest probability. So this is your reference penguin, or this is your experimental data you have collected, and try to align these three orientation. Obviously in this penguin in up direction it has a maximum, so you're assigning maximum. And when offside is down, roughly it match to some extent. So you give, maybe it can be like this or like this, but you don't clearly define it at initial state, this is the orientation. It goes with the maximum likelihood with the iteration as we follow, right? So this is how you will find a peak, then keep on doing this all process. And it goes in an iterative process. Your reference image, you have cross correlation, and then initially you roughly aligned. These are your experimental roughly aligned. Then use this better aligned image as a input or as your reference image, right? That's why we do this iterative process, 25 cycle, 50 cycle, 100 cycle. That's what we are giving iterative, that final orientation will be the input for the next round of, and you try to maximize its orientation. After some time, you will find, okay, all penguins are aligned nicely. Okay. Now coming to the 2D classification. 2D classification is same, what I just discussed. Instead of taking as is one reference, you take more than one reference, right? So suppose this is one reference, this is another reference, then you try to find this particle, whether it belongs to this, it belongs to this, and try to align in 2D. It goes in iterative cycle, like suppose you gave a 4 reference images, you have some data, and it will try to classify these, putting these in this orientation, this sometimes this will be noisy. You see, after iterative processes, the orientation of this image, it improves like this. And we also followed the same strategy. This is for a ribosome structure, where you could see after 25 iteration, we could see clearly ribosome particle, lot of ribosome particle, noise. These may be the degraded particle or something that auto-picked particle gave. So basically at 2D, you select the classes which shows the features, at least of your structure. So you may have some idea that, okay, my structure should look like this, and try, or you see that there is more than one classes that is showing the similar kind of structure, maybe a little different. So most likely this is your structure, and discard all these things. Okay, this is 2D. And if you take very noisy data, right, and give a reference, and it goes sometime to the reference bias. That's a classical problem that's called Einstein from noise. And one should be careful when you are dealing with the noisy data. It will, whatever you reference you give, it will try to make your structure like a reference. So careful when you are not very much sure that you doesn't end up with Einstein from noise kind of situation. Now, this is up to 2D refinement. But in 3D now, there is another variable, because you are putting in that 3D. 2D is in the 2D plane, so right, X by, you keep on rotating X by direction. But now you have the Z direction also. So how to align in a Z direction, or it's, so basically our 2D projections, the EM images are the 2D projection of a 3D object. And this is called the projection slice theorem. Before Saikat also described this. So I will just revise or refresh the thing. So from 2D to, how to go from 2D to 3D. Suppose you have a duck and you put that duck into the microscope. You get a 2D projection of a duck like this in different orientation, maybe some 100 orientation. Then you do a Fourier transformation of that duck. You get the Fourier slice that has a phase and amplitude information. You get a Fourier slice. You take a low resolution duck or a homogeneous duck, maybe rooster, and use that as a 3D model and do the Fourier transformation of that. It will be a 3D Fourier transform of the duck or your reference. And then try to align these 2D slices, and all the slices will be the central side. They all will pass through the central section of the 3D duck. So that is the basic, because 2D slices, they will be always passing through the center of the 3D Fourier transformation. And it was first time given this projection slice theorem by David DeRosier and Arne Klug. Basically, they use this bacteriophage tail like this one from one micrograph, because it has 32-fold symmetry. So it was easy to do the 3D reconstruction of a symmetrical object, because you know the orientation in the in the in this helical reconstruction. And this is kind of the first Cryo-EM structure of 3D Cryo-EM structure, we must say it, although it was very low resolution. Okay, so same projection theorem. What you do is in 3D, you have a real space, the 3D object, and for this you get the 2D slices and a 2D projection here. You can go back and forth. 2D projection, 3D projection theoretically, and these are the slice, and you align into a Fourier space in a 3D Fourier space, and it will always be a central slice. This is basically the 3D alignment. Basically, you have the initial you have these experimental images, and you have the reference 3D object, and you try to find out the best orientation or best position in its 3D object. That is how you do 3D refinement, 3D. Okay, or we can understand. This is a very nice review, and I would recommend to student to read this. You will get a better understanding of. See, this is your 2D projection, right? Setup 2D projection, and you got a 3D object, and you can make computationally its 2D. That will be a theoretical model projection, right? From here, like this, A, B, C, D, E. Now you take first projection, try to align it, either cross correlation of maximum likely. Here it is a cross correlation. Okay, so you tried first, and it is matching with the A. You tried second, it is matching with the E. Fifth is also matching with E. So you assign the orientation for these projection like A, B, and then you do a 3D reconstruction by assigning this, because here you know from a model that where its orientation is. So what you get is you get a improved 3D reconstruction. That's what actually the computer does. When it is cross correlation you are using, it is well defined. But when you are using maximum likelihood, it is telling that probability of projection 1 matching with A, B, C, D, E. It will not assign exactly that, okay, it belongs to this projection, but it will put a probability function over there. And with different, you start initially like this, make the theoretical projection, you try to match your experimental projection, where it matches, try to assign, and you get improved feature. With the different iteration, it keeps on going, and you finally, it converges, and you get a 3D map. If you look a 3D EM map and X-ray map, they look more or less same. Okay, so we reached somewhere, means 3D classification and 3D refinement. These are basically same. 3D classification, you are using more than one references, but in 3D refinement, it is the only one reference you are using. Okay. Now, because the particle, they undergoes a radiation damage, although it is imaged at a low resolution, but there is a lot of radiation damages going on. So this is called particle polishing. That what you could see is you can estimate the B factor, and how much is the B factor. Based upon the B factor, how much you should use that image in your movie refinement. One is this, the particle polishing. Second is, because initially I said that there's a movie correction that we do, that we segment our 2D micrograph into small pieces, then try to align and then stitch them together. But the particle, they are moving randomly. They are not moving in a coherence. So applying a correction to a micrograph may not be the ideal, but you need to, at later stage, you need to find out for each particle how that has drifted over the data collection time or as the correction time. Okay, that further improves the quality of your map. Once you have applied, this is called the particle polishing and Bayesian correction, CTF correction. In CTF correction, you apply the advanced level of this correction. Initially, it was right. I will not go to that more detail. Once you have that thing, because your data, it is aligned overall, but there may be heterogeneity because particles, they are fallen randomly. There may be heterogeneity because of factors occupancy is less, or because particle has a freedom to rotate, tumble freely in the ice, right? So another method one can do is the signal subtraction, where suppose your factor is this red, and it is bound to some big complex, and you are interested to find out this one, what is the confirmation of this. You want a high resolution because of we can say low occupancy, you are not getting high resolution. What you can do is you segment your factor into two, means your 3D volume, factor of your interest, that is this one, and the surrounding area. The total map will be your surrounding and your factor of the interest. Its 2D projection will be like this, right? It is a noisy, and if you subtract the region that is not of your interest, that is shown in yellow, if you subtract, you will get the 2D projection that has the 2D signal for mostly for your factor, and surrounding is less. When you try to align this image with this one, actual image, because of its extra region, this yellow, it will add up as a noise. But if you subtract, it will look like this. So obviously, you can better align this 2D projection to this compared to this. This is another powerful technique one can use to sort the heterogeneity or the occupancy. Now, another thing is, as I said, the particle, they are obviously mobile. There is flexibility, and there's another method that is called the multi-body refinement. Because if your complex is multi-domain, and all the domain, they are moving randomly. So when you try to align, the bigger domain will be aligned, but the minor, small domain, they will get blurred out. You could see clearly here, the center is aligned better, but there is a lot of blurring here. Instead of aligning this full particle, one can treat this as an individual body, this as an individual body, this as an individual body, and this as an individual body. Okay, and then do all these things, and you get a better result overall domain confirmation also. And for more, I recommend this paper from again Shore's group and Nagai's group. Okay, now once you have the structure, you need to validate. First thing is you visually you can validate, that is the best. Say your map is 9 angstrom. You should see the alpha helix. Your map is like 4.5 to 5 angstrom. There you should see separated beta sheets, because the distance between two beta strand in a beta sheet is 4.8 angstrom. I think 4.5 or you should see. If you don't see, it means that map your structure as a problem that may be because of this domain motion, heterogeneity, or the partial occupancy. Another method, I don't think nowadays people does this, is the tilt pair. You tilt it and then half of data you find out the pair. This is a little bit too, you check the handedness. Obviously, once you have high resolution, you can do this. And you see your particle and avoid this Einstein from noise issue. Now I will spend 5 to 10 minutes just to how we have used Cryo-EM, obviously the most powerful technique at the present, to solve the structure of 70S ribosome with a raw fetch complex at 2.8 angstrom resolution. See, ribosome, it's a protein synthesis machinery. It has 2 subunits, right? They join together and does its function. But it's a target for nearly 40% known antibiotics. And that makes studying, obviously, protein synthesis is a fundamental process and target that makes it too exciting to understand. What happens is ribosome, they hibernates. They go for sleep when there is a stress. They don't like stress. Okay, so how they do this thing? There are in E. coli and other bacteria, 2 ribosomes come together by a ribosome hibernation this promotion factor. Through its CTD dimerizes and they form a dimer. That is 100S. 100S can also be formed in a stress by, that is helped by 2 factors. One domain, this is a 2-domain protein. This is a single domain protein with another protein. It forms a 100S. If it is a single domain protein, it forms 70S. But in E. coli, in Mycobacterium tuberculosis or Mycobacteria, has 2 ribosome hibernation promotion factor. One is MPY, that is 2-domain protein, and another is RAFH protein. RAFH, it is only present, it is induced only in hypoxia and it is only in Mycobacterium or actinomycete, it's not in other, that makes it. But these are both a 2-domain protein. Ideally, it should dimerize and form 100S, but it forms 70S. That was the question we want to address. I will not go to much detail. I will go just come to here how we have successfully used this technique. This was the data collected in National Cryo-EM facility in CVS Bangalore. Initially, we did screening in our facility. As we have screening system, it's 200 kV microscope, also high resolution we collect from Bangalore. This is initially micrograph. We did motion correction, CTF refinement, particle picking, then picked these nice 2D classes. And we started with some 12 lakh particles. Then we did 3D classification based on maximum likelihood. And we found that one class has a eTRNA. There was a RAF bound, and there was a RAF, our factor bound with eTRNA. So because this is huge complex and our factor is very small, tiny sitting here, we thought, okay, we will do a focused classification. So this large subunit, we just masked it and focus only on the small subunit. We could see that the factor was actually in 47% of where it could begin see because of this large subunit artifact in alignment, right? So now it is a 47% particle as a factor. And then we did the CTF refinement and did the particle polishing. See, one thing important to note is our resolution was 3 angstrom. It was 2.9 and 2.8. Resolution wise, there was not much gain. But you see the quality of the map. That is the important. You see here it is fragmented, or maybe it was heterogeneous, not aligned. After this, you could see the quality, overall quality of map has gone up. Now our NTD, it was even flexible. What we did, we focus only this NTD of RAFH, and it's interacting components, this helix and this protein, and kept or subtract the signal for only these component from rest. So it looked like this. And the 2D classification, and we got better result density with another protein, or here it is only CTD, or it is A site tRNA. Because it's a small subunit and large subunit, and we know that it has an inherent movement. So we did multi-body refinement. Multi-body refinement means you fix this large subunit and give the rotation for the small subunit and align the small subunit. This way, its overall quality has improved. And here we did a low pass filter to 3.5 angstrom because still the factor was not that great, but we could. But here, what we did, we merged all these class, and finally we got the factor, because here we merged all three classes and got factor at 2.7 angstrom, 2.9 angstrom. So this is the power and how the low occupancy, if there is a flexibility, you can deal with nowadays on a computer, okay? And this is one should check the quality of map, the Fourier FSC, Fourier shell correlation. Basically, when you do a 3D refinement, you split your data into 2R and refine them independently and see that how the Fourier shell are correlating. At low special frequency here, obviously correlation will be highest, so it is close to 1. And as the resolution improves or special frequency goes up, the correlation between two will fall down. And this is called gold standard criteria. It is 0.143 cutoff, where if below that you don't take. So that defines your resolution in Cryo-EM. Another important thing is you should look at how your local resolution is, whether it is uniform, right? In our case, the resolution of the core is 2.5 up to 2.5 angstrom. And well, we can say up to 4 or 4 angstrom maximum, right? It means there is not too much flexibility or heterogeneity. If you have very heterogeneous map, you will find core is 2.5 angstrom, periphery is some 8 angstrom. Then you need to deal the heterogeneity again, okay? This is what structure we got finally, and you could really see that secondary structure resolved. Not only secondary structure, side chains are clearly resolved. And this is all done here, a data collector. I think it was an animation. I don't know what happened. Okay, yeah. Yeah, you could really see each and every amino acid nucleotide is resolved. I used to call it as it is like crystal clear, right? So obviously the EM, it is a powerful Cryo-EM, it's a powerful technique, it's an emerging technique, and it's a right opportunity. Youngster, definitely they must grab it, just grab this opportunity. You see each and every side chain nucleotide resolved. There's no ambiguity, nobody can question that, no, that was not right interaction. For drug discovery, or you need resolution kind of like this in vitro design, right? The CTD, now comes our question. CTD, after doing all these effort also, still resolution was not that great, but we are confident that the residue, they interact with the ribosome, we could see the side chain. What we found is it is a kind of a dimer kind of a structure, and it adopted the same dimer that the CTD or HPF long would adopt upon dimerization, right here. So it cannot further dimerize, so it stabilizes in 70S. So what we propose is, if CTD is repeated, it cannot further dimerize. The ribosome will be hibernated in 70 years. If it is a single domain, one repeat, it dimerizes. This is from the literature. This is also literature. If it is a single domain, another protein helps in forming a 100S. If it is a single domain, it remains 70 years. So this is what we added to the literature from this study. So this addresses this question because it's a larger and repeated, it cannot further form a dimer. But what is the problem with the MPY? It is nearly same. So this is what we would like to address in future. We applied the same strategy. I will not go to a detail to another ribosome. Here we formed the triple helix, and you see 3 nucleotides, they are coming on a same plane and forms a RNA triple helical structure. And our resolution is quite good enough that we can nicely see that obviously these are 3 nucleotides coming together. This is really very interesting and fascinating thing that we could see these nucleotide in resolved. And further for students especially, these are the very good reviews available and must read. This is from Sibel's group, very nice, nicely elaborated. And this is, I'm sharing the web link, and you will find really interesting, much interesting talk than what I gave just now on learning Cryo-EM. Grant Johnson's homepage, you will find this. MRC has 2 workshop. Just hook up and read, listen these videos. Steve Lutke's very nicely elaborated this 3D reconstruction, and New York Structural Biology Center frequently organizes different workshops. With this, I would like to acknowledge my hardworking student. The work I presented is mostly carried by Neeraj Shivani. Ankita works on, she also got very high resolution structure. Somin just joined, Thanjavur and Sadi, they are working on different aspects. RCB, faculty member, RCB administration, road grade, he gave us a plasmid. Our ATPC, especially thanks to Reena and Madhav. Reena, you have heard this morning. IBTC, where we do a lot of data processing. Special thanks really to Vinod Kumar and Sucharita from NCVS Bangalore. They are maintaining really a world-class facility in National Cryo-EM Center and funding from all these resources. With this, thank you very much for your kind attention. I will be happy to take questions. I can stop sharing.
Yeah.
Okay. Yeah.
Audience, if you have any questions for Dr. Prem, please. You can unmute yourself, or you can unmute yourself, Sulagna Haldar, and you can ask the question. Or you can post it on the chat. Sulagna Haldar, you raised your hand, so you can unmute and please ask your question.
Thank you for the nice presentation. I want to say, I want to gratitude to
Hello.
organizer. Hello. Hello.
Or you can post it in the chat box.
Open.
Prem, I'm Tyagesh here.
Yeah.
I just have a quick question. I'm just wondering, actually, there was a very nice and enlightening talk as usual. So I learned a lot of like insights into the Cryo-EM. I'm just wondering, actually, like, is there any kind of a research gaps in terms of like image data processing which you can let the crowd know, actually, or maybe which we can also fill in the gaps, like in terms of data picking, that image processing, which you might have encountered during your journey of Cryo-EM?
See, there are, I think, improvement in all that's going on if you see the initial CTF correction, it was just CTF correction. Now it is CTF refined. What I feel is, because biological sample, they are heterogeneous, right? They are not static because they have to perform a function. That's why there's a lot of motion going on. Dealing the heterogeneity and the low occupancy with the, if it is a complex, it is not still fully addressed. What is happening is, means people are generating 3D data from, say, 5 lakh particles. And if heterogeneity is not dealt properly, the program will say that it is 3 angstrom. But if you don't see the side chain properly at 3 angstrom, or you should start seeing at 3.5 angstrom or 3.8 angstrom, it means it is not the correct resolution, right? It is forced. And there I have seen quite a few papers. The resolution is claimed high. If you look their map, there is no feature. It means the problem of dealing heterogeneity is still not properly dealt. Maybe it's hard to deal heterogeneity, right? Like, means to generate a 3D 3D volume, it is easier than to deal the heterogeneity. I think to dealing with this heterogeneity, a lot of things need to be required. Yes.
Okay. Thank you.
What is the drawback of the technique?
Yes.
Okay. There is a question, Sulagna. What is the drawback of the technique? Well, every technique has a drawback. One is that it has the this preferred orientation, right? People are trying to do, Nikhil also elaborated on preferred orientation. Still, it is not dealt. Problem is, if your particle is flat, it will fall like this or like this, right? There is no mechanism you can put like this. One is, it depends upon this one. Second drawback is the radiation damage. We are using low dose. We are using a lot of, obviously it is in cryo, then lot of correction. Still, you will find that there is a radiation damage. How you will find that? You look the Cryo-EM map, you see the side chain of especially glutamic acid. You don't see the side chain. Means the COOH of the glutamic acid is not there, most of the map. That is because of radiation damage. It is not that, means you see arginine this much long side chain, but glutamic acid is not there. So radiation damage, preferred orientation are the problem, but that doesn't stop it from determining a high residue if somebody deal nicely. Yep. Yeah, now I can hear you. You can speak a little louder. Somebody asking question?
No, I don't think that was a question.
Okay, I see.
So just my curiosity, so you said there is radiation damage, particularly this glutamic acid. So how do people deal with it? I mean, that the confirmation is important, right, for understanding the biology.
No, people, how they deal, you take, when you are doing at final stage, this particle polishing, okay? You see that based on your B factor, after how many frame B factor is abruptly going, you stop there. So what is happening, although we are collecting in a movie mode, after some time, you don't see much information. By that time, the sample is totally burned out, right? That's why you stick to 20-25, even within 25. So if you want to, if your glutamic acid is a part of catalytic site, you use say 15 movie frame. Don't go to all 25.
Right.
That
right.
is the one thing.
Okay. Thank you. Thank you. Audience, do you have any?
Just one quick question, actually, in terms of like getting used to this data processing thing. Just wondering, like, can you please advise, we do have a bunch of students at NIT, SASTRA, and also we have an audience. If someone's really interested in getting used to this data processing technique, what is your taking it actually? Like, can they download the Relion and 3D Spark software? And is there any data availability? Is there actually which they can use it to train themselves?
Yes, very good question. So, see, I moved from X-ray to EM. So I also started at some point as a new. So you see, I am sure about Relion. I don't know the Cryo-Spark. Relion, every package comes with the tutorial. Okay. So that has a raw images and the processed data. So best way to start with is you download that data and just reproduce that tutorial, that you are getting the same number of particles, you are getting the similar 2D classification, you are getting same 3D classification, and you are getting final resolution same, that has a same similar FSC and local resolution. Don't do anything. So once you go through this process, you know that what are the steps and what I should see in each and every steps. Then you take a subset of your data. I see, problem is if you have 5 terabyte data and if you are a learner, it takes a lot of time on computer. By that time you forget, you start doing something else, right? So take a small data set. I'm 100 micrograph is more, 100, not 100, 20 micrograph, good 20 micrograph is enough. And do every step for one round from particle picking to the final movie polishing, etc. Because it is quick, so you are engaged, it doesn't take days, so you don't do anything, you don't start anything else. So that is the way I think that one should learn. And there is a CCP EM bulletin board. Just enroll for that. There is from last, I think from 2014 or 16. Whatever people have asked the question, they are there in archive. You have a problem, you ask a question over there. That is also a good thing.
Okay. Okay, Dr. Prem, thank you so much.
Thank you.
Yeah.
So, if there are no more questions, we will thank again Dr. Prem, and thanks for joining us and giving us a very enlightening talk. So we'll break for a short break and resume at 4:15. I hope all of you will be back by 4:15, and let us stick to the time. Thank you. Thank you very much, everyone.
Thank you.